and I'm starting to get nervous. So let's calculate something. Why is it that you should be surprised by the idea that cells can respond deterministically to 10% differences in concentration? So what are the physical limits to measuring concentration? It's a molecular signaling. So ultimately, right, you don't, if you look at a nucleus and you say, well, there's a concentration of molecules in the nucleus, you're standing outside the nucleus and you're pretending that you can count all the molecules inside. But there's no mechanism for doing that. The molecules have to act by some process. They have to arrive somewhere where they do their thing. So as it happens, we know that these molecules that are relevant for the process that we were talking about in the fly, the place where they have to arrive is some particular site along the DNA where they trigger or stop some process. But let's abstract away from that. Oh, and I should tell you that this isn't the first context in which this question arose. The first context in which this question arose was in how bacteria sense chemical gradients and are capable of swimming up the gradient so they find their way to food and away from molecules that they don't like. And there's this spectacular paper by Howard Berg and Ed Purcell from 1977, which inspired everything that came afterwards, okay? And their conclusion, just to give you, again, right, the theme is about the precision of biological function. Their conclusion was that the precision or the reliability with which bacteria can find their way up the gradient, up very shallow gradients of desirable molecules, that precision or reliability was so high that they must effectively be counting every single molecule that arrives at their surface. Because the precision, the difference between going up the gradient and going down the gradient was of the same order as the randomness in the arrival of the molecules. Really, it's a beautiful argument. And that if it's true that they're counting every single molecule, you should be able to demonstrate that directly, and eventually that was done and so on. Okay? It's also true that the bacteria are so small that according to these calculations, they can't actually measure the difference in concentration between the front and the back in order to measure a gradient. The only way they can measure a gradient is by averaging over their volume and checking how things are changing over time as they swim. And so that means that the whole strategy they have for navigating in a chemical gradient is determined by the physics of the noise in counting molecules. Okay. By the way, it also means you should be able to fool them, right? Because if you just pour more stuff into the container, they should think that life is getting better and keep going in whatever direction they're going because they think things are getting better. And that experiment actually works. Anyhow, so what's the argument? I have a little detector, and it's of some linear size. Maybe it's this binding site along the DNA where the protein has to find it. So effectively what I'm doing is counting molecules in a volume whose linear size is A. Okay. Well, how many molecules are there in there? Well, on average, it's the concentration of molecules times the volume. Well, that's because that's what concentration means, right? It's not a result. That's the definition, right? But the important point is that if the molecules are at low concentration, then they're effectively not interacting with each other. And so that means that their actual positions are random. So if I have random points in space, and there are C of them per unit volume, then if I take a volume A cubed, on average I count n bar is C times A cubed, but I will also have fluctuations in that count, And the variance of those fluctuations will be the same as the mean, right? Because it's a Poisson process. So that means that there will be fluctuations which are of order the square root of this quantity. But in the same way that concentration is defined as the average number, you could say that if you count, you could divide by the volume in which you counted and get an estimate of the concentration. So if you say your estimate of the concentration is something like n over a cubed, then your fractional error will be the same as your fractional counting error, and that'll be 1 over the square root of the mean. But that's if you count once. So you look inside, and you see 50 molecules, and you say, okay, the concentration is 50 divided by a cubed. Can you improve your estimate by counting again? Well, if you count the same molecules again, that doesn't help, right? I mean, unless you got it wrong the first time. So let's assume you can do it right. But you know that the concentration is the same in some larger volume. And if I wait a little while, the molecules in this volume will diffuse out, and the molecules in this volume will diffuse in. And if I wait for this volume to re-equilibrate with the environment, then I'm counting a new set of molecules. So how long do I have to wait? Well, I have to wait for a time, which is the time it takes to diffuse in and out of this region, which is the diffusion, which is the size of the region squared divided by the diffusion constant. Or maybe it's better to write it like that, right? If I wait for a time tau c, then with diffusion, I'll move over a distance of size a. And so if I wait that long, I get a completely independent sample. But you know that if you average together independent samples, you should lower your noise level by 1 over the square root of the number of samples. So you should be able to do better by a factor, which is if you're willing to average for a time tau average, then in that time, you will see tau average divided by tau c samples. And so you get an improvement, which is 1 over the square root of that time. So now, you plug this in, and one of those beautiful things happens where all the factors averaged. They all line up in a very nice way. And you get that your fractional error is 1 over the square root of the diffusion constant times the size of your detector times the concentration times the time that you're willing to average. So let's think about this for a moment. You all realize that if you average for a long time, unless there's something unless the correlations in the noise have some long tail or something, which of course can happen, but in this case probably not, then as long as you're diffusing in 3 dimensions, not in 1 dimension, so you're diffusing in 3 dimensions, then the fluctuations should have short-range correlations in time, and that means that your quality of your measurement should improve as 1 over the square root of the time. So that should be here. You believe that if the detector is bigger, it should do better, right? Because you're counting more molecules. So there should be something related to the size of the detector. It's not obvious that it should be linear. But you also know that you're counting. So the more molecules you have, the better you should do, and it should again go like 1 over the square root. So if you put these 2 factors here, you realize that what you're missing is a volume and a time in the denominator. And this combination is exactly what you need in order to get the dimensions right. So this is almost the only thing you could write down by dimensional analysis in 3 dimensions. There's another way of thinking about this answer, which is to draw parentheses. If I give you some of you may remember this if you've taken a chemistry course. If I have 2 molecules that have to find each other, there's a rate at which that happens. That rate is proportional to the concentration. And so there's a second order rate constant, which, if you think about it, has units, well, it has units of 1 over concentration, which is to say volume per unit time. And if you make a very simple model of a chemical reaction, which is that I have a blob of size A, and I have another thing diffusing around, and whenever I hit the surface of the blob, the molecule disappears, and that's the rate, right? That's the reaction. Then the problem you have to solve is I have a spherical surface, and that surface has concentration 0 at the surface, because every time a molecule hits it, it disappears and reacts. And then I have diffusion in the bulk, which has to meet this boundary, 0 concentration boundary condition. And the only way I can meet that is to have a flux at the surface. And you integrate the flux, and that's the rate constant. Right? That's the rate at which the reaction's happening. If you do that problem, this is the answer. So this is the rate at which new molecules are arriving inside the volume of size A. But they're arriving at random. So this is the rate. This is the time over which you average. So this is the number. So this is just 1 over square root of n. This is exactly the same as the argument that says that if you're trying to form an image, the accuracy with which you measure intensity is 1 over the square root of the number of photons that you count. Here it's 1 over the square root of the number of molecules that you count. In the case of the photons, they arrive as a Poisson process, either because they're coming from a laser and that's a property of coherent states, or because you're only collecting a very small fraction of the photons, in which case the ones that you get are a random choice out of the original stream. And in both cases, you get Poisson statistics. Here it's the same Poisson statistics, but for a different reason, right? It's because of the properties of the random walk, but same idea, okay? So this is just counting statistics, and you can't do better. So there's 2 directions now. One is, should you believe this hand-waving argument? Can we do it? Actually, this is why the Berg and Purcell paper is so interesting. By the way, some of you may know, I guess this is a generational effect. For how many of you is the name Purcell familiar? Yes, there's certainly a generational effect, but good, okay. So some of you may know him as the author of an electromagnetism textbook. Others of you may know that he led one of the 2 teams that discovered nuclear magnetic resonance. Okay. Yes?
You mentioned the Berkeley course specifically.
Sorry?
You mentioned the Berkeley course specifically.
Yes, yeah. So right. So if you read the Berkeley physics course, the electricity and magnetism book was written by Purcell. But the reason they asked him, right, was because he discovered NMR. It's kind of important. Anyhow, so he was one of. Anyhow, there's a very interesting story behind all this. Anyhow, so Howard Berg is the guy who discovered that bacteria swim by rotating their flagella. So he discovered the world's smallest rotary engine. And when it came time to think about this, he went back to Purcell. Purcell also wrote a very well-known paper about the motion of bacteria, which is called Life at Low Reynolds Number. Spectacular, same period. Anyhow, so this paper is remarkable because it's filled with these kinds of intuitive arguments. And for everything they want to do, they have some clever thing where they say, oh, this is like that, and so I can do this, I can make this estimate, and so on. There is not what, I never had the chance to talk to Purcell about this, but I have talked to Berg about it. There's not a single honest calculation in the paper, right? It's all intuitive. It's also, as far as I know, all correct, okay? But, you might say, one thing, when you arrive at this point is, can I do it, before I start using this argument in many different situations, you might like to do a real calculation that gave you this as the answer rather than just waving your hands. Okay, so that's one thing. We'll do that because, this is a school, so we should, and as I say, I spoke for too long without calculating anything, so we'll do a real calculation in just a moment. But let me put in some numbers so that you understand why this is important, okay? So, numbers for the control of gene expression. So, this whole process by which the information that's encoded in the DNA gets read out to messenger RNA to protein is called the expression of the gene. And as I say, this happens, one of the main ways in which this happens is that protein molecules called transcription factors bind to the DNA. So what is the typical concentration of these molecules? It's measured in nanomolar. And let me remind you, this is 10 to the minus 9 times 10 to the 23rd molecules per liter, which is totally useless. It is 0.6 molecules per cubic micron. That's useful. So at this level, right, you have no business carrying around the 6. That 6 is this 6, of course. What's the diffusion constant? Well, it's the diffusion constant of a protein inside the cell. That's measured in microns squared per second. I mean, go look, okay? Do the experiment. What's the size of the detector? Well, there's this piece of DNA, and here's the part that codes for the protein. And there's this big blob that has to march along and spit out the messenger RNA molecule. And then there's some place here where this molecule comes and binds. How big is that? Well, it's the size of the molecule. It's maybe 3 nanometers. So let's say the detector is 3 nanometers, which is 10 to the minus 3 microns. So D times A times C is 3 times 10 to the minus 3 times 1 times 1. So 3 times 10 to the minus 3 is 1 over 300, and the time is in the denominator. So delta C over C is 1 over the square root of DAC, which is tau times tau, which is tau divided by 300 seconds. But you'll remember that 300 seconds is 5 minutes. So this tells you that if these numbers are right, if you have to make decisions in minutes or tens of minutes, you're going to have trouble measuring concentrations to within a factor of 2. So maybe some of these numbers are wrong. But remember, and they're probably not exactly right, and we could talk about that, but I don't think that's the important point. Remember that in order to have delta C over C be 1/10, so that you can get to 10% accuracy, then the number inside the square root has to be 100. You certainly don't have 500 minutes in which to make this decision. Okay? 500 minutes is nearly 10 hours. It's a little less than 10 hours. And remember that in 24 hours, the animal will walk away. Right? All of this is happening in the space of 10, 15 minutes, as I showed you that boundary, right? So you're really up against the wall. So this is a hint that the system, if put some numbers in front here to change things around a little bit, you're not going to escape completely from this problem. Okay? What this means is that the ability of cells to respond to 10% differences in transcription factor concentrations is in a neighborhood where the fundamental physical limits of counting molecules are actually close to the performance of the cell. So, if that's true, maybe we can take the idea that the system approaches these fundamental limits and turn that into a theory of how this network in the middle works, right? Or at least how the information gets read out at the end. So if you want to take seriously the idea that the system pushes as much information as possible out of this limited number of molecules, then there are many steps where you think you could make predictions. So one thing is, you really better be sure that you don't add extra noise on top of this. So various mechanisms that are involved in this process of reading out the genetic information had better be kept quiet, as it were. Okay? They shouldn't add too much extra noise. There's another thing, which is that in some sense the reading is being done by this network. And that means that there's some inevitable noise that comes along with this, and you might want to be sure that the concentrations that you use, so for instance, if this network turns on when the concentration is up here, then it won't do anything no matter what the noise level is, right? It'll just always be off. So that's obviously stupid, and you can't imagine you'd find the biology working that way. But there's a more subtle version of this, which is that you'd like the way in which this network and the individual elements in the network respond to be such that they actually move through their full dynamic range as these concentrations move through the range that they do at the input end. So in order to be sure that you get the most information out of a limited number of molecules, there has to be some match between the range of signals at the input and the characteristics of this network through which they're going to propagate. Right? Remember, in this network, there are lots of little devices, each of which has its own input-output relationship at every node, right? And you run the danger that the distribution of concentrations at the input to the node won't be matched to the place where the input-output relationship is sensitive. And so you can improve how much information you transmit by making all those matches work. There are all the parameters inside this network that can be used to help you transmit more information. For example, the fact that the different elements in the network turn each other off means that together, which is true, together they produce timescales which are longer, right? Because there are feedback loops in the network, you get emergent timescales in the network which can be much longer than the timescales of that operate at the individual nodes. Those longer timescales give you longer averaging times. And we know that that's helpful for suppressing the noise. On the other hand, if they're hooked up in the wrong way, it's possible that through that feedback you would amplify the noise. So there's a certain amount of design that you can imagine goes on inside this network. The thing which I'll actually talk about, because we've made progress on it relatively recently, is what happens at the very end when you have to read this information out. So by the time you get to here, there's a small number of different molecular species in this network that are carrying all this information about position, and you might guess that there's pressure to read that information out as precisely as you can, again, to get as much information as you can out of a limited number of molecules. So I'll talk about that, but before I do that, I want to at least sketch for you why we think that the Berg-Purcell argument is correct, okay? So let me do a real calculation, and then we can discuss its generalizations. So molecules binding and unbinding, in the first approximation, they come to thermal equilibrium. So what's happening? What's happening is you get a change in the concentration of the molecules in the surrounding solution, and then at this particular place along the DNA, there's a binding site, and on average, that binding site increases or decreases its occupancy, right? And that's an equilibrium process if you wait long enough. If that's true, then your errors in measuring concentration arise because the occupancy of the binding site fluctuates in time. And so what you want is a theory of how the occupancy of the binding site responds to changes in concentration, and a theory of the fluctuations in the occupancy. But this is an equilibrium process. The fluctuations in occupancy, although you might think of it as some stochastic process, it's really just an example of thermal fluctuations. So let's do the theory of thermal fluctuations in the occupancy of this binding site. So how do I do that? Well, if I imagine I have many copies, right, I sort of coarse grain a little bit in my mind, I can talk about a variable n, which would be 0 if the occupancy was, if the site were unoccupied, 1 if it were occupied all the time. And because I'm going to coarse grain, I'm going to write a differential equation for this, even though it starts out being a discrete variable, right? So So what I have is, if the binding site is empty, then there's a rate at which it gets filled, which is K plus, remember that rate constant I was telling you about, the molecules finding their target, times the concentration of the molecules at the position of the binding site, which I won't worry about just yet, we'll get back to it. And then, of course, if the binding site is full, it's possible for the molecule to fall off, and that happens at some rate K minus. So everybody's okay with this? Good. Now, I want to do a theory of the response to changing concentration, and I want to do a theory of the fluctuations. So since I have thermal fluctuations, you remember that the way in which you compute thermal fluctuations is through the fluctuation dissipation theorem. And the fluctuation dissipation theorem tells you, for example, that the power spectrum of fluctuations in x, where x is some coordinate, is 2kT divided by omega times the imaginary part of the response function, the linear response function of position to its conjugate force. Right? This is preparation for a statistical physics conference. You remember this, right? So if you want the theory, and alternatively, you can think of this as the position responding to a fluctuating force. Whose power spectrum is related to the imaginary part of the inverse response function. Remember that when you invert, right, 1 over I is minus I, so you need a minus sign here, okay? So what do I need to do? I need to so the first thing I need is, well, what coordinate do I care about? This coordinate is going to be n, right? That's the thing I'm watching. I'm watching the position, the occupancy of the binding site. It's like watching the position of a Brownian particle. But what's the conjugate force? Well, remember that if you take a coordinate and multiply it by a force, you get an energy. Right? So this, I mean, eventually this has to go back to mechanics, right? So what's the energy that you multiply by this? Well, it's the energy difference between the occupied and the unoccupied states. But where is that energy difference in this equation? Well, it's there because of detailed balance. You have K plus times C divided by K minus is e to the energy difference divided by KT. And this energy difference is the conjugate force. Okay. So how do I compute the response to changes in this conjugate force? Well, I imagine that K plus and K minus fluctuate. And when they fluctuate, they produce a change in delta E. So let's linearize this equation around some average. Right? If all you have are fluctuations in n itself, then when you linearize, you get a term from here and a term from here, and those fluctuations will relax exponentially with this rate, which is the combination of these two rates. If the concentration itself changes, then that shows up from this term. Okay? But then if the rates change, you get a term which is plus, let me write it this way, K plus C 1 minus n bar delta K plus over K plus, right? So if this rate changes, then that produces this term, and then you get K minus n delta K minus over K minus. But you'll notice that these n bars are determined by setting n dot equal to 0, right? That's the average value, or the constant value, the steady state value. And at steady state, this term and this term have to be the same. and you'll notice that you get the same term. So that means that all that matters is delta K plus over K plus minus delta K minus over K minus. But if you look at this, you can convince yourself that this is just delta E over KT. Right? If I take this equation and do all the variations, right, I get delta K plus C divided by K minus delta K minus K plus C over K minus squared is equal to delta E over KT, e to the E divided by KT. And so here you take one of these K minuses and you put it here, and here you put a K plus here and a K plus here. And now you notice that you have these common factors, and so these all cancel. And this is the combination that you need. So the result is that this is saying that delta N dot is minus K plus C plus K minus delta N plus K plus 1 minus N bar delta C. And you get K plus C 1 minus N bar delta E over KT. Okay? So by taking this part of the equation and this, I can figure out what the linear response functions are. So in particular, well, so there's two things to notice, right? One is that by comparing these two terms, what you see is that a fractional change in concentration is equivalent to a change in energy divided by KT. So if I can compute the strength of this fluctuating force, just like I do in the theory of Brownian motion, right? If I can get the analog of the Einstein relation that tells me how strong the fluctuating force is, then I will know how big my my errors in concentration are. Because I need to change the concentration by an amount which is big enough to overcome these fluctuations in the force that's acting on the system, right? In the same way that if you're watching a Brownian particle and you want to know whether you applied a force or not, you need to apply a force which is bigger than the Langevin forces that act over the same amount of time. So it's exactly the same calculation. Okay? So you can do this, right? And in particular, you can see that delta E, if I just Fourier transform this, delta E over delta N, right? There's a delta E on this side, there's delta N there, there's a KT, and there's a K plus C 1 minus N bar. And then you'll notice that here there's a minus I omega from the derivative, and here there's a K plus C plus K minus. And since I only care about the imaginary part, right, this tells me that the power spectrum of the fluctuations in E looks something like 2 KT divided by omega times the imaginary part of this thing, which is KT divided by K plus C 1 minus N bar. And there's an omega here, which cancels the omega, and that minus sign cancels that minus sign. And what you get is just KT squared 2 divided by K plus C 1 minus N bar. And because of this relationship here, if you ask about the fractional noise in concentration, it's just this. So this is the power spectrum of an effective fluctuation in the fractional concentration. So that tells you that the thermal fluctuations in the occupancy of the binding site produce a noise whose power spectrum is such that if you want to change the concentration enough to get bigger than that noise, you need concentrations whose variations themselves are this big in their power spectrum. And if you average for a time tau, then your bandwidth of frequencies that you're sensitive to is 1 over tau. So you get this times 1 over tau. Okay? Great. The only problem is this doesn't look anything like the Berg-Purcell result, right? This has rate constants k plus in it. It has 1 minus n bar. What's going on? And the answer is, well, one way to think about it is that k plus is a rate constant, so it's limited by diffusion. The noise is smaller if k plus is larger, but it can never be faster than d times a. Right? Because that's how long it takes for the molecules to diffuse up to the site. So you could say the answer is that if you just replace k plus by its diffusion limited value, you'll get back something which is always bigger than the Berg-Purcell result. And so that tells you that the Berg-Purcell result is a lower bound. In fact, you can do a more sophisticated calculation than that. You can actually say, well, I left something out here, which is that I'm only sensitive to the concentration at the point where the detector is. And the concentration itself is varying in space because it's diffusing. And whenever the molecule binds to the detector, it gets pulled out of solution. So at that point, I lose a molecule, right? If you now couple these equations together, what you'll find is that the response function grows a new term, which is the back action, right? When I take a molecule, well, one way it's easier to think about it this way. When the binding site releases a molecule into solution, it can diffuse around and come back. And that slows down the dynamics. And that slowing down of the dynamics lengthens the correlation times, which means that the noise level in any given bandwidth goes up. And so what happens is if you solve these coupled equations, you end up adding a self-energy here. Well, I guess it would be more conventional to put a minus sign there. Okay, so for some fraction of you, you know why I'm using this language, right? And for the rest of you, it doesn't matter. Right? Just do the calculation, okay? What you'll discover is that when you couple these things together, you get back a term here. And this term is the result of the coupling to diffusion. And in fact, if you work it out, you find that as omega goes to 0, this term goes like minus I omega, or plus I omega, times the integral over a Fourier variable of dk squared, right? Because again, you need to find the response of the concentration to this drive from n, and then you evaluate it back at the same point where the sink was, because that's what counts here. And what you find is something that's inversely proportional to the diffusion constant. I told you it acts like almost like an inertia, so it basically multiplies this I omega term. And it's nominally divergent in the ultraviolet, right, at short distances. And so you should cut that divergence off at the length scale of the receptor itself, because that's the smallest scale on which you trust this whole picture. You said that the detector was a point when you wrote down this equation, and that's not exactly true. Presumably it has some linear dimensions. So this whole description should be cut off at a length scale of size A. When you do that, and you go through this whole calculation, you discover that there's an added term to this spectral density, and that term is exactly the Berg-Purcell limit. Okay? So the point of this rather short discussion is not that, well, it's a fun calculation, okay? It's an exercise. So usually you think about thermal fluctuations, you think about voltage fluctuations in a resistor, you think about Brownian motion. Remember that chemical systems also have thermal fluctuations. If they're in thermal equilibrium, the fluctuations in the states of the molecules, including the species, right, the concentrations themselves, those are thermal fluctuations. Indeed, the square root of n fluctuations in diffusion, those are thermal fluctuations. The coordinate is the concentration. What's the conjugate force? The conjugate force is the chemical potential, because that's the thing when you multiply by the concentration, you get a free energy. So if you compute the response function of concentration to a change in chemical potential and work your way through the fluctuation dissipation theorem and average over a certain volume, you'll get back the square root of n fluctuations. Maybe that's not the easiest way to find that answer, but at least you understand that these are all connected to each other, okay? But importantly, you can generalize this calculation to more complicated schemes. What happens if I have binding to many different receptors that are near each other? Well, then the fluctuations in the concentration of the different receptors become correlated, because when the molecule comes off of one receptor, it can rebind to another by diffusion. And this strategy allows you to take account of those correlations automatically, which Berg and Purcell did, again, by brilliantly waving their hands. You get the same answer that Berg and Purcell got, by the way, because they really understood what was going on, even if they couldn't do the real calculation. You could then let those binding events become cooperative with each other, which we know happens a lot in biology. Turns out you still get the same Berg-Purcell limit. Cooperativity affects the analog of this term, but not the added term that comes from diffusion. So by following this strategy, you can convince yourself that the Berg-Purcell limit really is a much more general limit than you might have thought from the original qualitative argument. Okay? And sometimes that's the power of sophisticated theoretical methods. It's to convince you that the thing that you wrote down intuitively really is the right answer and that nothing goes wrong. Sometimes you do the calculation and lo and behold, the thing that you thought was intuitively right doesn't work because, for example, some correction diverges. Right? And we all know examples of this that were very important in the history of physics. But sometimes you get back what you thought was right, except now you know you're right. Okay? And so that's how I think this more sophisticated statistical physics gets used here. Okay, tell me when I should stop. I should stop in 15 minutes? 5:30. Okay, good. All right. So let me pause here and ask if there are questions. It's allowed, you know? Yeah, there's one brave guy. You guys should follow his lead. Go ahead.
Actually, it's not about this whole calculation.
Okay.
There's also other processes, like you're talking about fluctuations around 10%.
Yeah?
But cells also have like signal propagation, signal traffic.
Right. So what's important here is that when you think about this limit, let me go all the way back to the intuitive argument. What the intuitive argument tells you is that the limit to your ability to measure is set by the rate, average rate at which the molecules arrive at their target. And the reason that's such an appealing intuition is because then it doesn't matter what happens after they arrive, right? So they can trigger some very complicated non-equilibrium process. Signals can be amplified. They can be fed back. You can do all sorts of things, but you can't get around the fact that you can't measure signals, you can't count molecules that didn't arrive. And if they arrive at random, then there's some error associated with that counting, and all you can do is count for longer. You can argue a little bit about the factors of 2 out in front, which might be important, but not at this level.
Another thing that this diffusion process is considering that only these molecules are not, because there's a crowd process.
Right. So another calculation you might want to do is, what if this molecule, as it's moving around, can also bind to other things? And that slows down. So it has 2 effects. One is it slows down the diffusion. So I suddenly have to worry, is the diffusion constant which matters the bare diffusion constant or the diffusion constant after I take account of these other effects? But the other effect, of course, is that if the molecules can bind to other sites, then the concentration of molecules which are free to actually bind to this site is lower than the total concentration in the neighborhood. So you now might ask, which concentration do I use? But the interesting thing is that the only thing that matters is the product. And in simple models, the product doesn't change. The amount by which the diffusion constant goes down is the same as by the factor by which the free molecules are reduced relative to the total molecules. So if you take the total number of molecules and the observable diffusion constant, you'll get the right answer. But this deserves more thought. There's also a question of how you measure the diffusion constant,
Yes.
right?
The theoretical diffusion is totally different.
So you have to be sure that the diffusion constant that you measure. So I think the way to do it is to set up models for what you think is going on, both microscopically and in terms of your measurements. And then ask yourself, what's the relationship? Remember, you have to worry both about how you measure the diffusion constant and how you measure the concentration. Okay? Because probably, if you paint all the molecules with a fluorescent indicator, then you're measuring the total concentration. You can't tell which ones are free and which ones are bound, right? So you're measuring something bigger, you're measuring a bigger concentration than the ones that are actually available to bind. And then the diffusion constant you measure is normalized downward, but by an amount that depends, again, on exactly how you're measuring it. So there is a concern here. So you have to be careful. I think by now we know the answers, and this is not, those things are all true, and you should be careful about all of them, and they have implications for how we think about this whole system. I don't think they're the answer to this problem. I think that's the fair statement. Okay, good, that's a good problem. Yes, somebody else maybe? Okay, you get another chance in 15 minutes. All right, so what's the point here? The point is that it really is true that something like the Berg-Purcell limit is relevant even in this complicated situation. So this is an example, in some sense, the role of more sophisticated theory here is to justify the use of something that seems incredibly oversimplified in a real context. Because this calculation, you can keep doing more and more complicated versions of it, right? And you keep getting back the same answer. So you say, okay, now I trust the answer. This tells you that these limits are very relevant to the real system, and I think that's a generally very important conclusion. But now we want to know, can we use this in some way to think about how the system is constructed? So let me just take one moment. When we were over here with the sort of philosophical background, what I emphasized was that one of the things that drove the change in attitude about the physics of biological systems being part of physics as opposed to the application of physics to another field is a change in the quality of the data. So let me take a few minutes to tell you how you can reach in to the fruit fly embryo and actually measure concentrations accurately enough that all the things that I'm talking about have a chance of making sense, okay? It's not a complete guide to the experiments, but I think it's important that you get some sense. So there are a couple of references up here. So I should say, was it somebody once said there's no history, there's only biography? So my involvement in this happened around the time that I moved to Princeton. A colleague, David Tank, who's a brilliant experimental physicist who spent most of his career thinking about how to record signals in the nervous system, and actually I'll talk about some of his experiments tomorrow, because we've continued to collaborate on these things. Both of us had been thinking about problems in signals in the brain, and we were interested in how the same conceptual questions played out for signals at the molecular and cellular level. And literally, as we were talking, trying to figure out whether there was something to do together, David was very interested in new imaging methods. I was interested in these theoretical issues of the flavor that I've been telling you about. Our colleague Eric Wieschaus walked into the room. And as I told you, Eric is one of the heroes of the story of the fruit fly. And David and I tried to explain to Eric what we were talking about, which was difficult because literally we didn't know what we were talking about, right? We were trying to figure out whether there was something to do. And Eric smiled and very graciously said, well, I'm not sure what it is you want to do, but let me explain to you why you should do it in flies. And he proceeded to explain all the advantages of the fruit fly embryo as a laboratory. And he ended by saying, and if you have a physics student who wants to try doing these experiments, send them to me and I'll take them in the lab and show them whatever they need to know. And one important part of this story is he did not say, I'll have one of the postdocs show him everything he needs to know. He said, I'll show him. So my student, Thomas Greger, who'd come to Princeton to do theory, got wind of this story. And he said, well, I don't know if I ever want to do experiments in my life, but at least I'll be able to tell my grandchildren that some Nobel Prize winner took me in the lab and showed me how to do them. So he did. And well, long story short, Thomas decided to become an experimentalist. He ended up with 3 advisors, me, Eric, and David. He figured out how to work with 3 advisors, which might actually be the hardest part of this. You know, 1 experimental physicist, 1 real biologist, 1 theoretical physicist. I mean, we obviously all have different views of what's interesting and important. And at the end of the day, Thomas had to decide for himself what he wanted to do. And he did a spectacular job. He's back in Princeton as a professor. He's also some fraction of his time at the Institut Pasteur now in Paris. And since coming back, he's been on this steady march to try and measure everything that's going on in this system more and more accurately. So let me just give you a snapshot of the kinds of things these guys can do. So one thing you can do is you can take the embryo. So this is the same view as we saw before here, right, where you're looking down on the embryo in a slice this way, right? A slice through what's called the mid-sagittal plane, which is a fancy word for the fattest part. And what you do is one way to measure concentration of proteins is to stop the action, which you do by gently cooking the egg. You then take the protein that you're interested in and you inject it into a mouse. And the mouse notices that there's something foreign, so it makes antibodies against the protein. You purify the antibodies. Then you take a goat, and you take antibodies from a mouse and you inject them into the goat. And the goat notices that there's something foreign, so it makes anti-mouse antibodies. And then you attach a fluorescent marker to the anti-mouse antibodies. So now you take the embryo and you bathe it in the antibodies against the protein that you care about. And those antibodies stick to that protein. You then bathe the embryo again in antibodies against antibodies, except those are fluorescently tagged. So now if you measure the brightness of the fluorescence, you should get something that's proportional to the concentration of the protein that you started with. You realize that this sounds more like cooking than like a physics experiment. Not just the cooking the egg part, but all the rest of the mixing and everything, right? So you should cook but not scramble, right? Because you want the spatial pattern to stay fixed. So you have to do a lot of experiments to convince yourself that the fluorescence that you measure is proportional to concentration with an accuracy of a few %. And there's a variety of ways of doing this. For example, you could genetically engineer the fly to make a fluorescent protein and then make antibodies against the fluorescent protein. and be sure that that the fluorescent antibodies have a fluorescence that's proportional to the intrinsic fluorescence of the protein you were targeting. Okay, it's one of many experiments you have to do. There's another thing you have to do, which is that when you stop the action, you need to know when you stopped it, right? Because if I have a cup with 100 embryos in it, they didn't all start time 0 at the same time, right? The mother lays eggs. So I do my best, right, to collect embryos from mothers, 20 minutes, 30 minutes, 50 minutes, whatever, an hour point, this time after the eggs were laid, but it won't be exact because the eggs don't come out at the same time, right? Insects lay lots of eggs, right? This is one biological fact you need to know in order to understand how these experiments are possible. So you need a clock. Well, one clock is cell division. At time 0, there's one nucleus. After some time, there are 2 nuclei. Then there are 4. Then there are 8, and so on. Everything that we're interested in happens when there are 2 to the 13th nuclei, most of which are now at the surface of the embryo. And so this is all happening in cycle 14. The reason I was careful to say nuclei rather than cells is because flies are special. This is all happening so fast that the nuclei duplicate, but the embryo doesn't build membranes to separate them into cells. That happens during cycle 14. So all these nuclei are near the surface, and the single membrane that's surrounding the entire egg, which remember started as one cell and is now one cell with 2 to the 13th nuclei, that embryo starts to fold in. That membrane starts to fold in, so at each point that's needed so that it can become the beginnings of the membrane that separates the nuclei into separate cells. If you look under the microscope when the embryo is live and you follow the progress of that membrane folding in, you find that if you do it in multiple embryos, the progress of that membrane moving is a clock for time during cycle 14, which is accurate to better than 1 minute. So this is another example of incredibly precise behavior. In this case, it's not a subject we're going to investigate. We're just going to exploit the fact that there's this underlying precision of the timing. That means that if you stop everything and you want to know when did it stop, just measure the length of this advancing membrane. And that'll tell you when you were in cycle 14 to 1 minute accuracy. If you remember that all these concentrations are changing in time. So if you don't have a clock, you'll have 2 embryos that have different concentrations, and you'll think that's just noise. Okay? It's not. It's changing time. So you can then develop antibodies against several different molecules, and these are the 4 different proteins that are in this network in here that I was telling you about. The more modern way of measuring concentrations, and all these molecules have names, by the way, and I'm going to see if I can avoid using the names. There's A, B, C, and D. And you can see that these, you started with these input concentrations, which were very gradual along the whole length of the membrane, of the embryo. Here you have things that are more localized, but still don't obviously look sharp enough to make these stripes. So somewhere in here, the information must be hiding. There's a more modern way of measuring protein concentrations, which is to genetically engineer the fly so that whenever it makes the protein, it has a fluorescent protein attached to it. That's great because then you can watch everything happen live. It's hard to do because many of these processes are very fast, and the fluorescent proteins take a long time to settle into the right structure where they're fluorescent. So your images of the fluorescence are probably lagging the concentration. So I'll show you one for demonstration purposes. By the way, if you want to measure this concentration, that's great, because this molecule, remember, the messenger RNA was put in by the mother, and it gets translated, and so it lives for a relatively long time. And by the time you're looking in cycle 14, it's had plenty of time to mature. And you can see the fluorescence very clearly. And for example, that's how you can show that the absolute concentration is reproducible from embryo to better than 10% accuracy, just by taking two embryos side by side and measuring the fluorescence. The fluorescence is the same to within 10%. But here's an example where, I hope, there we are. Thomas and his colleagues genetically engineered one of these striped things to be fluorescent. And there you see the stripes coming in real time. This is seen from the side. You'll notice that the width of a stripe, you see the discrete cells, right? So the stripes, once they sharpen up, are only a few cells wide. So that at least gives you a hint of the underlying precision. If you don't like measuring protein concentrations, you can try and measure the messenger RNA concentration. What you can now do is you have a messenger RNA molecule that you care about. It's thousands of bases long, or 1,000 bases long, let's say, right? If you're 300 amino acids, that takes 1,000 bases, or 900. So I can take a little piece of DNA, which is complementary to this segment of the messenger RNA. Maybe it's 20 base pairs long, so that it'll be unique. There will be no other sequence in the entire animal that looks exactly like this. So if I diffuse this molecule in, it will find all these messenger RNA molecules. But now I can take this little probe and I can attach a fluorescent protein, a fluorescent marker to it. And I can use a small dye molecule, right? Because I'm synthesizing this. I don't need the animal to make it. But since this thing is 1,000 base pairs long, I can take these 20 bases and these 20 bases and these 20 bases and these 20 bases and these 20 bases until I have 25 different things that I'm hitting, right, to make 1,000. Actually, that makes 500. So I have even more space. But every single one of those has a little fluorescent molecule attached to it. But now I have 25 fluorophores in the same spot. If I look under a fluorescence microscope, I can see the fluorescence from 25 molecules that are in the same spot with no difficulty. And if I'm very careful, I can localize the position of those extremely accurately. What that means is that I can count every single messenger RNA molecule of that species in the entire animal. So if I'm interested in messenger RNA molecules, I can turn the problem into one of counting, okay? And I get ultimate precision. There's a difficulty, which is that if the messenger RNA molecules become abundant enough that they get close to each other, remember, concentrations are usefully measured in per cubic micron. If the concentration becomes several per cubic micron, then the typical distance is less than 1 micron and becomes comparable to the resolution of your microscope. And that means that you could not tell whether there was one molecule there or two. Unless, of course, you trust your measurement of the intensity. Because if there are two molecules, it should be twice as bright. So as with many things that we do in physics, you use pulse height discrimination in order to distinguish different classes of events. But that means that you have to have control over all these things, right? So this is to give you a sense of what's possible, okay? And by now, this is all very well developed, and you can measure not from just one mRNA species, you can measure from many, you can do it in different colors at the same time, you can wash things out and use multiple colors, you do all sorts of stuff, okay? And this whole world of making antibody measurements, making fluorescent proteins, counting messenger RNA molecules, this is all moving, right? So all these technologies are advancing. And this is an example of why you can ask questions today that 10 years ago you would not have been able to trust the existence of quantitative measurements. Okay? So let's talk about, maybe we can turn down the lights in the front. Thanks. Let's talk about this last step. I've told you that the concentration differences here are so small that they're close to the physical limits that come from counting every single molecule. I've told you that if that suggests that the whole system is organized to squeeze out as much information as possible from a limited number of molecules. If that's true, then there are many, many things that you might think the system has to get right. So let me tell you about one of them. This is the problem of decoding the information which is present in the output of this genetic network. So I told you that in this network, in the middle of the embryo, let's say between 10% and 90%, okay, so don't worry about the 10% at the edges, which is hard, there's several reasons not to worry about it, so don't worry about it. If you look in that middle 80%, there's really only 4 of these molecules. And we can measure the concentrations of all of these molecules. So if I'm a cell sitting somewhere, how do I know where I am? Well, the information is contained in these 4 concentrations. So how would I get it out? Well, suppose I could only use one of them to start with. So let's pick this one. You'll notice this is one whose concentration peaks in the middle of the embryo. So if you measure the concentration, you'll see that it goes up and comes down. The particular blue line is from this picture, and then the gray that you see in the background is the probability density, right? It's the probability that you'll see a concentration of this size at this point. And you'll notice actually that the fluctuations are relatively small. They're not as visible on this plot as I would like them to be, but they are very small. So the fluctuations, right, on average, if you do it in many embryos, you get 1. This particular embryo, you got up to 1.2, but that's actually an outlier. So fluctuations are perhaps 10% in concentration at this point, smaller along here, okay? But this is what we see as observers, right? At every point, we see a mean and we see a variance. The cell has this concentration to work with and has to figure out where it is. So you should sort of turn the picture on its side, right? If I'm a cell sitting here, I see this concentration. And what do I know? Well, it might be that well, let's do the easy one. Suppose I see a really high concentration, then the answer is I must be somewhere in the middle, right? Because that's the only place where the concentration is high. If I see an intermediate concentration, then I might be on this flank or I might be on that flank. If I see a low concentration, the only thing I know for sure is I'm not in the middle, but I don't know whether I'm somewhere in this vast region of the front or the back. So conceptually, what you're doing here is you're using Bayes' rule, right? You measure the probability, or you infer from measurements on a large number of embryos, the probability that the concentration or gene expression level will take on a certain value at each position. And then you construct the opposite probability distribution, P of x given g, and you'll remember the basic rule. So P of x given g is P of g given x times P of x divided by P of g. All right, that's Bayes' rule. And in this case, it's easy because the distribution of x is where am I? The answer is I'm somewhere between 0 and 1. Right? That's all I know for sure. It's uniform, right? The cells are uniformly spaced. So this is a constant, and if you think about it, this plays a role, is independent of x, right? So it just plays a role of normalizing this probability distribution. So really, P of x given g is just proportional to P of g given x if you think of it as a function of x, which is why you can just turn the picture on its side and you get this. Now this is very nice, but it's very hard to draw this picture if instead of using 1 gene, I use 4 genes. Because I would need 4 dimensions. I need 4 x-axes. So what am I going to do instead? What I'm going to do is, let me imagine that I'm a cell whose actual position is x. Given that I'm a cell whose actual position is x, I happen to see this concentration of this molecule. I then look up from here what positions are consistent with this measurement. And I plot that density along this axis, right? So what this says is that if I'm a cell who's at this position, then typically I will think that I'm either here or there. If I'm a cell that's here, I would think that I'm pretty much where I am, but with a big error bar. And if I'm a cell here, I know absolutely nothing about where I am except that I'm not in the middle. Good. If this was the whole story, then, and it's useful to remember that since probability distributions are normalized, the height of the peak is related to the width. So if I show you the density, the peak of the density, which is what's on this axis, right? How dark is the gray? Darker gray means that you have a higher peak, which means you have a narrower distribution, so you have greater precision in determining position. So now, instead of using 1, let me use 2. And because the second gene is not symmetric in the same way, some of the ambiguities are resolved. And then there's a third gene that sits in this no man's land here where you didn't know anything, and that resolves some ambiguities. And then finally you add one more, and it sits in the space in here that resolves the last of the ambiguities. And now you see that if you are a cell that is sitting here, you're pretty sure you're sitting here. And if you look at the height of the peak, you notice that it has gone above 100. And so that means that the width is less than 1%. So these 4 molecules contain enough information to specify position to 1%. And remember that the stripes are specified to 1% accuracy, and the furrow that forms the boundary between the head and the rest of the body is also specified to 1% accuracy. So fantastic. It's also true that the error is pretty much uniform along the whole length. And I told you that there should be some matching between the concentrations at the input and the noise levels. And there's an argument to be made that that constancy of the error is a reflection of this matching. So that's very nice. But how do you know that this is actually how it works? In fact, we don't. All we've shown you is that if the system uses all the information which is available from these signals, that is sufficient to reach 1% accuracy. But is that what the embryo actually does? Well, I've hidden from you exactly what function of concentration you're supposed to compute, right? In here, I've shown it to you explicitly. I tell you how to get from the concentration of this molecule. Sorry, I've used its name, but I won't say it, or the abbreviation of its name anyway. I've told you how to get from this concentration to an estimate of position, but as I start using more and more molecules, I've hidden that calculation from you just because it has too many axes. But in order to construct this picture, I must have that calculation somewhere. So if I gave you any set of concentrations of these 4 molecules, I could tell you where that cell believes it should be, right? So let me use the power of genetics in the fly to manipulate the system. And I told you there were 3 input signals, one at the anterior end, one at the posterior end, and one at the terminals, so A, P, and T. And let me start knocking them out. So if I knock out one of these, things look like this. If I knock out the one in the front, predictably it has big effects in the front. If I knock out the one at the back, it has big effects at the back, although strangely they spread toward the front. If I leave all of them in place, this is what it looks like. If I knock out all of them, I get nothing, right? Everything's just flat. That's a good thing because it tells you there are no other sources of information about position. You can also knock them out 2 at a time. Amazingly, if you have only the signal from the front, you still get a lot of structure for these 4 genes. So the question is, can I actually, for instance, take one of these, and I'm a cell sitting here, I could take the level of the green molecule, the level of the blue molecule, the other 2 are 0, and feed it into my calculation and say where that cell thinks it is. There's a technical point which I'll skip over. So let me do that for the case where I knock out the terminal systems. Let me also say that this work, I told you that this whole project began with Eric Fischhaus and David Tank and myself and Thomas Greger as a student. He's back as a professor, and one of the first students that he had in his lab was an undergraduate, Mariela Petkova, who's now a grad student at Harvard. And she did all of these experiments and actually was the one who convinced us that we should listen to Eric and study mutants. Because, of course, mutants have been very good to Eric. He won a Nobel Prize with them. But as a physicist, I was always very suspicious. Mutants seem to me like a sledgehammer, right? You're literally, they use the phrase, knock something out, right? That's a huge perturbation. So you might worry that with these huge perturbations, you just take the system as a way of showing that that molecule is involved in the process. Fantastic. As a quantitative tool, I was very suspicious. The technical part I'm skipping over is why it works, why you haven't knocked the system as far from the real system as you might have expected, but that's a longer discussion. So let me try to finish up and tell you what happens. So I told you what we're going to do. We're going to take the calculation that we did in the normal embryo, which tells us for every combination of these 4 molecules, where do you think you are? What's the probability distribution of positions? And we're going to construct that for this mutant. And when you do that, you see this ridge of probability that actually tracks the diagonal very well and then veers off. And you see that the effects are primarily at the front and at the back, which makes sense because what we knocked out was the thing at the 2 ends. So the middle is fine. How would we know if what the embryo is doing is doing the same calculation that we're doing? Well, you see, if you think you're here, then if you were a normal embryo and you were there, you would make a stripe. This is one of these molecules here. So you would make this stripe. So since you think you're here, you should make a stripe, and you do. And similarly here, you're off only by a little bit, but that's enough that you should put the stripe here, and you do. And there are more examples like this. This is one of my favorites. Here's where you knock out the thing in the front, and now everything goes haywire. You get a segment of the map that looks sort of reasonable, except it's displaced, but then the stripes are also displaced. If you look carefully, you find that in some embryos, there's density in this region, and in other embryos there isn't. And so that means that sometimes you should see stripes here and sometimes you don't, and that's actually true. In a normal embryo, you never see missing stripes or extra stripes, but in these mutants you do, and in exactly the place that's predicted. And the favorite one is this one, which says that there's this density here, which is a cell that's sitting in the front and near the head, but it should think that it's near the tail. So it should make a duplicate of the 7th stripe, and it does. And importantly, if you dig into the details of what's happening along the DNA, you can tell the difference between the regulatory events which lead to the production of stripe 1, stripe 2, stripe 7, and so on. So you can show that this is actually a duplicate of stripe 7 and not a shifted version of stripe 1, exactly as we predict. And okay, you can do more of these. This one's kind of fun, right? And that's it. So what I hope to have shown you is that the idea of the extreme precision of these systems is something that we can push on in several directions. There are theoretical issues about where the boundaries are, right? So I tell you the system is very precise, but relative to what, right? So in absolute terms, a 10% difference in concentration, well, it was enough to make the biologists think it was impossible, but we don't know to begin with whether that's really something we should be impressed by or not. So there's a theory of what the limits are, and that theory can become more and more sophisticated, and it's interesting to push in that direction. But then there's the idea of turning this around and saying, if the system is approaching the physical limits, then a certain amount of how the system functions should be determined by the fact that it's pushing the physical limits. And so we've given you this example of how the information that's encoded in this intermediate network gets read out to make the stripes. And of course, there's a complicated molecular process by which that happens. And so to go back to the things you heard this morning, there's this issue in thinking about the physics of biological systems, how much detail do you need to think about? And the answer, of course, is that we never know for sure. What this is showing you is that we have a theory of how these stripes move around in response to mutations at the input, which makes absolutely no reference to any of the molecular details. The only thing it uses is the idea of reading out the information as efficiently as possible, and that produces a quantitative theory, which is accurate at the level of 1% in the positioning of the stripes, which is the precision that the system really achieves. So the reason that I find this work exciting is that it reaches a level of theory-experiment comparison, even in mutants, which we would not have thought possible 5 or 10 years ago. And so we may or may not be right about the theory, okay? One has to be, I'm old enough now that I can say, I would like to be right. If it turns out that we're not, it's okay. At your age, you really still need to be right, right? You got to be right that first time. okay? After that, you can relax, okay? But seriously, we might have the wrong theory. But now you can't propose a theory that doesn't connect with experiment at this level of precision, because that's where we are in being able to do that kind of comparison. And it's at the level that we're used to in physics, which wasn't true just 5 years ago. So thanks for your patience.
Can I ask a
Yes.
very naive question? So you had a back of the envelope estimate here for a single concentration. And then you were showing that from that very simple estimate, it's hard to imagine such high precision. Is there a simple way of but then you show that if there is more than one concentration, then this gets better, right? Is it a fair estimate? So can I put some 3 squares somewhere there? 3 concentrations?
Well,
is there a modification of the simple argument with more than one?
So this is why I think it's important that when you do the back of the envelope calculation, you get something that's way off in the bad direction. So that tells you that there's a problem. The question of exactly which factors get put together in order to resolve the problem, I think, is still open. Okay? I mean, we did, in the paper where we first did this, we had a first try, and I think it was a very interesting try. I don't know whether it's the right answer or not. For example, let me give you, just to jump all the way to the present time. We now know that in big complicated in contrast to bacteria, in big complicated organisms like us, and even in fruit flies, at the place where a lot of transcriptional activity is happening, there's often a collection of genes that are all being transcribed at the same time, and the binding events that regulate this are very distant along the DNA from, so this distance that I've sketched here is actually much, much larger. And the DNA is folded up in space so that in 3 dimensions, the distance is not as long as it would be in 1 dimension, but it's still substantial. So the picture that we now have is that which is partly based on direct observations, is that there's this cloud in which many, many proteins have condensed into a kind of transcription factory, or a little droplet. And the gene which is being transcribed is in there, and so are all the binding sites. And so now, maybe the way to think about this is it acts, if you go back to Bergen-Purcell's original calculation, if you have many binding sites that are arrayed around a larger sphere of size R, even though the individual binding sites are of size A, if there are enough of them, they act like one big target of size R. And in fact, that region is about 100 times this size, which would be just enough to solve the problem. So maybe that's the answer. We don't know for sure. The question of whether you have multiple proteins, remember, the proteins that I was showing you here are from in here, whereas the thing that you initially have to discriminate is this one. And there's certainly a region in which this is the only one you have to work with. Although I would not put my hand in the fire for square root of 3, right, at this stage.
Thank you. I'm amazed. About the experimental
Yes.
Side of the thing, when you attach the GFP to a target gene, a target
Yes.
protein, do these, like the protein would have a bigger volume?
Absolutely.
Yeah, and then it will
Great
induce
question.
like a diffusion coefficient to be lower. Something
Worse
like
than that. You have this big thing hanging off. How do you know it just doesn't get in the way of the molecule functioning?
Yes. So does it have
So-
an impact in the experimental?
Great question. So the really successful use of the GFP tag in this set of problems is for this molecule.
Okay.
I said, the first reason I said that was good was because it lives a long time, so before the things that you're interested in, so it has a chance to mature and give you the fully fluorescent molecule.
Okay.
The other thing is that you know from independent experiments that if you change the concentration of this molecule, you move the furrow that forms the neck. It's called the cephalic furrow. Sorry, a little bit of the jargon, okay? So if you made the protein and for some reason it didn't come out at the right concentration, well, if it doesn't come out at the right concentration, you can see it. Well, actually, you don't know. In the old days, when this was first done, old days meaning 10 years ago or more, when you wanted to put something back into the genome, you put it in at random because you really couldn't guide it very much. Nowadays, you use CRISPR and put it back exactly where you want.
Yeah.
Since you put it in at random, there are variations in the expression with exactly where it goes. And you can see that because you see differences in the fluorescence. That's actually useful because it means that you can control the overall concentration. So you turn your artifact into a tool. We also, however, you don't know which one of these is the concentration that occurs in the normal system. But what you do know is that if you change the concentration, you move the cephalic furrow. So there is one of the constructs where you put it back in where the cephalic furrow is in exactly the same place as it is in the wild-type embryo. That's the one you study.
Okay.
Because you have a functional readout. So I should say that you can use GFP tagging to tell you where proteins go, and then this doesn't matter, right? Because once they go there, you see it, you're done.
Sure.
If you want to use it as a quantitative tool, then you have to convince yourself that you have replaced the function of the normal protein, but only with a light bulb, right? But the only thing you've done is to add the fluorescence.
Yes.
And so you need to measure something to show that it's the same. So literally, you have two lines of flies, right? Two families of flies, one of which is the normal one, and the other of which has had exactly one gene taken out and replaced with that gene plus the green fluorescent protein. And if you look at the body plan, you look at the position of the neck, you look at the expression of these stripes, they're the same. To within the accuracy of the reproducibility of the embryos themselves. Okay. So you're confident that you can do that. But that's a huge amount of work. And that's one of the reasons why I think it's still true that as a physicist interested in biology, the set of experiments you want to interact with, you really want to be able to talk to them and know how calibrated they are, right?
Okay. Thank you.
Any other? So let's take a break. Thank you, Bill. Please, the groups that are going to present now, at 6 o'clock, start the presentations. Please test your computers. You are going to use your own computer or upload your presentation in the Institute computer. Thank you.